Let's look at Clostridium difficile. Antimicrobial resistance from phenotyping point of view. Toxinotyping, which is a PCR-RFLP method as we did before. And genotypic profiling, looking at the strain diversity that we did in swine a few years ago. This was part of my former student Pamela Fry's master's project, and I think it was published just last year. So, Clostridium difficile and see we've been doing, okay? Clostridium difficile, not safe pork importance, even MRSA, by the way. We are at safe pork, but MRSA is not really a foodborne pathogen. We can discuss that. It can be arguable. Some people may say Staph aureus generally cause food intoxication, but we have not seen MRSA strains from pigs being a food safety or foodborne pathogen. I'm not aware of that kind of report. Definitely not from pigs, but even in human medicine. I saw one report in hospital that food handler in the kitchen of hospital contaminated with MRSA was the patients. The patients are highly immunosuppressed and they got MRSA. But that's extremely sporadic reports that you see. But if you know, maybe tell me. But in retail meats they have been found, yes, but you don't see a foodborne disease necessarily because of MRSA. Clostridium difficile, the same thing. It's not a foodborne pathogen, but I'm giving you demonstration since we're dealing with molecular aspects. C. difficile causes, it's a gram-positive spore on all everything, but it causes C. difficile associated diarrhea all over the world now these days. It's becoming more and more important community-associated pathogen. What does it do? It causes pseudomembranous colitis that you see here. If you have the pathogen in the GI tract and in a healthy colon, it can cause pseudomembranous colitis that you see on the right-hand side on some situations. A little bit of the pathogenesis. I mean, why we do C. difficile is, of course, in piglets, we commonly see Clostridium difficile. In humans, it's more and more now community associated. It's been known as nosocomial before, but now community associated. Typically what happens is, Clostridium difficile is common in the environment and different surfaces. The spores are common, but when you ingest the spores, once it gets into this, it can pass the GI tract, mainly if you are taking proton pump inhibitors, gastritis or others, in a typical risk factor way. Once it gets to small intestine, it changes to vegetative state, mainly when it gets exposed to bile salts and other things. If it reaches to small intestine and changes to a vegetative state, then the production of enterotoxin and cytotoxin, if it is a toxigenic C. difficile, results in pseudomembranous colitis. But not all C. difficile are toxigenic. Some are non-toxigenic. Okay. So look, the strain is hypervirulent type. They release lots of enterotoxin and cytotoxin, which causes the pseudomembranous colitis. So that's an important problem. So proton pump inhibitor is known to be a risk factor to some extent, with additional other factors, exposure to the spores, then oral antibiotics, because inherently C. difficile is mostly inherently resistant to antibiotics, some key antibiotics. Okay. Now, so that being the in detail. Traditional risk factor, usually you get this pseudomembranous colitis if the person is an old age group, history of an oral antibiotic, because C. difficile is inherently resistant to many antibiotics. And if there is high gastric pH, proton pump inhibitor, other things that enables the spore to survive the low acidity, the low pH in the stomach. Those are the kind of traditional risk factors. Recently, there has been a history of, apparently healthy patients having C. difficile and coming into the hospital. No history of antibiotics, other things. Hypervirulent Clostridium difficile. So I'm giving you the history and the epidemiologic things as we prepare to your research, just like I gave you earlier. And increasing incidence, increasing severity, young age group, and many other these newly emerging risk factors have been reported about since 2004, 2005. Okay. Their units looking at the nosocomial infections started looking at different potential risk factors. What was the change? Number one, healthy persons in the community, not hospital associated, became infected. Sometimes younger age groups, not necessarily old age. No prior antibiotic use history, but they were succumbing to Clostridium difficile infection. So there was a question, what's going on with this? Is there a new strain, epidemic strain, circulating not just in the US, Europe was the same thing. In fact, right now, UK, I think, according to at least the BBC News, not scientific report, I saw C. difficile, hypervirulent C. difficile surpassed MRSA. So it's become a big issue. So what was happening now let's get into the molecular component. If you look at the genomic segment of Clostridium difficile, what makes the strains toxigenic? And then there's hypervirulent aspect. Generally, toxigenicity, Clostridium difficile has these 5 genes that encode for enterotoxin and cytotoxin. Okay, so now the point I want to make here is when you do molecular epidemiology of organisms, whatever epidemiology you want to address, before you launch your molecular tools, you have to have some understanding of the issue you are addressing. In our cases, the big question, I don't know if the question is already maybe cut down slides, is now with this newly emerging hypervirulent strains in humans, CDC was looking for a solution or a source. What is the source of these hypervirulent strains? National Pork Board was invited to Atlanta, and I was invited to go there and discuss with the US CDC, Pork Board, other people, and people from equine, people from other human areas, discussing how is this emerging hypervirulent strain, where is it coming from? We know that C. difficile occurs in piglets. Is it coming from pigs? They asked us, do you want to do a study? So there is some funding. So I said, okay, let's do it. Okay, even though I'm not really a Clostridium person. Dr. Glenn Sanger from Iowa, he was at Arizona at the time. He does lots of Clostridium difficile experts. He does in cattle, he does in other different, in pigs and others. But the question was, where was this, what's the source of these hypervirulent strains? So we started looking at what makes them hypervirulent. This is already reported before. Okay, this is the region in the chromosome of the organism. We need to understand that first. There is an effector gene, cytotoxin gene, TCDB, the enterotoxin gene, TCDA, and then there are some regulator genes. This is a positive regulator gene. If this is hyperexpressed, then enterotoxin, cytotoxin comes in. There is a negative down-regulator gene, TCDC, here. If this is defective, what happens? It cannot suppress the enterotoxin and cytotoxin, so the strain becomes hypervirulent. So what CDC was seeing in these multiple hospitals, Pennsylvania, New York, Ohio, Chicago, different hospitals with these patients coming from the community, was a specific strain. That's where now the molecular epidemiology comes into the picture. They found that it is toxinotype 3. They named it as NAP1, which stands for North American Pulsed Field Type 1. On ribotyping, which Europeans use lots of ribotyping, is ribotype O27. So that's the designation of this epidemic strain. But we don't know what's happening in pigs. Do they carry hypervirulent strain? If they carry, is it O27? Is it NAP1, toxinotype 3? So that's where the study is. If you don't do molecular epidemiology, just do the bacteriology, you get C. difficile. Of course we get from piglets, but you cannot just claim, because we get high prevalence of C. difficile in piglets, that's the source for the human patients. We need to do phenotyping and genotyping. So let me jump into the results. So we did a study in Ohio and North Carolina for pigs and humans from several farms and in piglets and sows, especially the first 2 weeks of age. We got up to even 75%. It's highly clustered. They are all together, but a good high percent. From humans, we got positive ones from North Carolina as well as Ohio. But here is the results. If you look at antimicrobial resistance profiling, you can see that both human and swine isolates are highly resistant to ciprofloxacin, close to 100%. Cipro is very important antibiotic, but remember, that is also the problem with C. difficile is it's inherently resistant. So just oral antibiotic taking ciprofloxacin, while it's it might be helpful for some pathogenic organism in your GI tract, it gives a chance for C. difficile to grow. It kills commensal organisms, many of them. C. difficile, which many of us may have right now, will be happy to proliferate and cause diseases. Okay. If you look at tetracycline, we found much more, close to 60% of the isolates from piglets, but not in the human, 10%. In terms of strain similarity, we already have some idea, right? So they might be different because the percentage of tetracycline resistance is different. Or there might be some similar, but many different. Erythromycin, there is some similarity, okay, as you can see here. And then just on human isolates, few but considerable, significant is vancomycin resistant. We have never seen vanco-resistant C. difficile from pigs. For hypervirulent C. difficile in humans, there are two big antibiotics being used in human medicine, vancomycin or metronidazole. So it's very bad to see really vanco-resistant ones. We had a couple of isolates that are metronidazole resistant as well, but we never seen a human isolate that were resistant to both metronidazole and vancomycin. That would be even more dangerous. Happily proliferates then cytotoxin, enterotoxin. If you are really young and fit, everything, your immune system will take care of it usually. But added host factor, if you are old age or immunocompromised, pseudomembranous colitis, perforation maybe, up to death. Okay, so it's a very serious infection. This is comparing from our study between the human isolates of human origin.
Big production here?
No. In fact, it's this globally, but definitely not in the US, never. In Australia, you guys used before. Europe used before avoparicin, not vancomycin, but avoparicin, a glycopeptide that has cross-resistance, has been used before, but since 1997, it's been discontinued. So nobody in the world really uses it. I don't know if in a clinical veterinary setting, if in dogs, cats, I know they use, but not in food animal production system. So we did some PCR. That's one initial step to look at what are the resistance genes. Do we have a similarity between the human isolates and the pig isolates? There are some similarities in one of the tetracycline resistance gene, but difference as well. Highly in the swine ones, 100% was TET-M, and one sub-carried TET-W, another gene. The human ones never carried this gene. So you can have some ideas as the strains might also be different if the genes are different, but we can't say for sure, and this shows you the different. Now the key step is starting from here. In terms of now, we already know what the human epidemic strain looks like in terms of the toxin genes. The toxin A, which is cytotoxin, toxin B, which is enterotoxin, these which are highly expressed. Now, what did we find from the pigs? A good number of them had both toxins, so they are toxigenic, 75%. About a quarter of them were not toxigenic. Some of them, but it was all Clostridium 1 or 5. But anyways, they were. There is a binary toxin. They had binary toxin, which its role is not very well defined, but everybody tests it. We tested it. Yeah. Now, when it comes to the down regulator gene, is the down regulator fully functional or is it defective? We know that the human epidemic strain, which makes it hypervirulent, the down regulator gene has 18 base pair deletions, so it's not functional. What do we get from the piglets? Interestingly, that one was also defective, but we found 39 base pair deletion. Potentially, that is hypervirulent strain. Okay, so now by doing the molecular, not only amplifying the gene, but sequencing the gene, we know that it's 39 base pair deletion, and this is the same, almost all of them were same with that deletion mutation. So the sequencing is important. The amplification is important. Molecular epidemiologic study. Another very interesting thing we found is, you remember the downregulator gene I showed you, TCDC? It had majority of the strains we did from pigs, more than 70%, had 39 base pair deletion. This is even larger deletion than that epidemic strain circulating in humans. So now, what we know so far up to this level is, we know that majority of the isolates from pigs are toxigenic. We could have done phenotypic assay, but based on the genotype. We know that they could be hypervirulent because the downregulator gene has a bigger deletion. We have fairly good indications that they are different from the epidemic strain. Why? The epidemic strain has a specific 18 base pair deletion of the downregulator. But we consistently got more than the 70%. All of those toxigenic ones had this 39 base pair deletion. Hadn't we done the PCR and sequencing, in this case, we wouldn't have known that, right? If we had done, for example, phenotypic assay, we may get some cytotoxin, enterotoxin, the human isolates, you get the same. It will be harder to differentiate. So we already have a very good idea. The next step we didn't discuss today as a methodology is called PCR-RFLP, restriction fragment length polymorphism. That's important for us to compare to answer our epidemiology question whether the pigs carry a similar toxinotype as the epidemic human strains. The epidemic human strain is toxinotype 3. That is across the board. In fact, United Kingdom also reported the same one, hypervirulent one, and they said even surpassed MRSA in the UK, according to BBC News. Not scientific, but I think it's enough and a trustable, wonderful news source to trust. So what did we find in our study from the pigs? There was none of them were toxinotype 3. So that clearly now answers that we don't have the epidemic strain in the pigs. Therefore, the likelihood of having pigs as the source of that toxinotype 3 to humans is not almost zero. There are lots of isolates that were tested. The predominant one for us was toxinotype 5. Okay? Number one, phenotypically we had some idea. The isolate seems to be different. Number two, based on the sequencing, the downregulator gene had a different profile. It's potentially hypervirulent, but a different profile. The deletion was much larger in the pig isolates. Now, number three, based on toxinotype, the epidemic strain is toxinotype 3. What we found was predominantly toxinotype 5, toxinotype 0. In the human isolates we got was either non-toxigenic and then toxinotype, non-toxigenic, toxinotype 0, or toxinotype 8. Some of the pig isolates had a mixed one. You remember the discussion earlier, pig 10 colonies, and that's always an issue. In Campylobacter, we see a mixed heterogeneity. We published that a few years ago and reported the safe work as well as published. Salmonella, we see, and now we pick Salmonella, we decided to pick 3. But we don't see that much variation, not like 5 or 10. But within 3, we even get up to 3 serotypes sometimes. Rare, but we see. Or phenotypically, or susceptibility, or other differences. So we picked 3 for Clostridium difficile. 12% of them had 2 mixed different strains in them. So there are some mixed ones. But at this stage, we knew that the epidemic strain, we didn't have it, at least in our study in Ohio and North Carolina, from pigs. So, CDC is more targeting in different locations. So just before in Clostridium difficile, I was wondering, we find this high percentage Clostridium difficile for the first 2 weeks. Then we go after a month, none of the pigs are positive. What's going on, Glenn? Are we wrong? Are we missing? Is this assay is a problem? It's not a problem. It's natural. And he told me that the host, some receptors in the intestine in the host, which is pigs, change, which makes it not so conducive to Clostridium difficile. Just so think about the host pathogen environment interaction. But we had a lot more from early age, and we had 24 isolates of human origin. So if you look at what toxinotypes we found, toxinotype 5 was the predominant in the swine isolates. But we didn't find any toxinotype that in the human isolates we got from our study. And we had, some of them had 2 types of 5 and non-toxigenic. Why? You remember co-infection, mixed infection? We picked 3 colonies per pig to see heterogeneity. We just discussed about heterogeneity in Campylobacter, right? You may have mixed infection between jejuni and coli. We had the same issue here in Clostridium difficile. Most labs, whether it's Salmonella, pick 1 colony. I recommend always to picking more if you have resources. Then you see some variation, like 12% of the pig isolates had more than 1 Clostridium difficile in their intestine. Toxinotype 5 and non-toxigenic ones. And then some, the same thing mixed for 4.6%. Some of them are non-toxigenic. The human ones, in this case, we have more data from this study, but these were clustered isolates from specific hospital, specific unit. So I'm not very much surprised, but it's not telling us the correct story. Now we got lots of toxinotype 3, the epidemic strain also we got on this study after we did it, but this was a little bit outdated data. So we have a lot more isolates. But the point I want to make is now you can do toxinotyping, PCR-RFLP, to be able even to differentiate, to genotype and be able to have a stronger indication that whether you have a clonal related isolates between humans and pigs. So just looking at so far what we looked at with the deletion mutation difference, with the, even though they are all C. difficile, they are all cytotoxin carrying, most of them, and enterotoxin, looking at the deletion mutation of negative regulators, the toxinotype difference, if you ask me, are the pigs, now if I ask you, do you think pigs are the primary reservoirs of this epidemic strain, which is toxinotype 3, with 18 base pair deletion, what would you say? Which one? Yeah, no, it doesn't look like that. Yeah, with a very large certainty. Then we did PFGE as well. I don't know, with their other collaboration using, and we did PFGE, that's confirmed the same thing. So it was not very unique for us. They did PFGE previously from humans and pigs in a different study. They found the same thing as we did. Toxinotype 5, they have binary toxin, and they found the same 39 base pair deletion. So that strain seems to be very common, and others report, I think Scott Weiss in Canada reported it as well. It's got high deletion. It's toxinotype 5 based on the PCR restriction fragment length polymorphism. It's a genotyping method by amplifying the toxin genes, and then you cut them using 3 or up to 4, I think it's 4 enzymes, and then you look at the profile, the binding profile. It's straightforward method. But they found some from pigs, and as well as they found it before in humans. But right now we don't have that as an issue, but it's important consideration. But the good thing is C. difficile does not occur post 2 weeks. After 2 weeks of age in piglets, almost we never find C. difficile. It's not an issue in finishing, it's not an issue in nursery. Just in the farrowing units we get high percent, then it disappears. It's not like the pathogen disappears, but the host, the intestinal receptors are not conducive. The intestinal receptors change as their age progresses, and that's not conducive for C. difficile, so we don't see it much. So really, that's all I have. I think that's the last slide for C. difficile. So we can. Okay, the question is: did you do sequencing after we do the PCR RFLP? Sequencing of which gene? The enterotoxin, cytotoxin?
Yes.
We did not.
How did you know that
39? Okay. We did TCDC. That's why I asked you again the question. Not the cytotoxin and enterotoxin gene. We sequenced the regulator gene. Is that what you mean? Your question is?
Down-regulator, yes.
Of course, that's why we know with the 39 base pair deletion.
So you sequenced it without?
Absolutely. We amplified it. First of all, we had an idea, but without sequencing, is there any other way to know I had 39 base pair deletion? How would you know? There is, at least I'm not aware of any other way. So what we did was, yes, we amplified it. We could have cloned it into a vector, but we already know the sequence of this TCDC gene. We designed, we purified it from the gel. And just also as an advice to you, if you already know the sequence, fairly sure, you don't need to clone it or create a library. It's not all, it's just one gene. After we amplified, the size we expected was lower. So we suspected something. Pamela, the student who saw this, she was curious. Wow, this, I expected if it was 18 base pair deletion, this level, it was not, a little bit slightly, but ours look a little bit lower. So she purified it, did everything, sequencing primer, we sequenced it. And then we get the sequencing. How would I know 39 base pair deletion? How would I know? Tell me. Just if I ask, this is a question. Not just Masfin, but any of you.
Well, actually,
How would you do, once you get sequencing data, how would I know I have that 39 specific base pair deletion?
amplified AF.
AFL, no, I don't need to go back to genotype. It can, well, yeah. No, it's more than AFLP. I already have sequence in my hands. It's, I think, easy question. I don't want you to think it's a difficult question.
I have a sequence.
You have a sequence. How would I know? What do you do? Compare. Good, okay. How do you compare? Where do you go to get that previous sequence reported in the world? NCBI BLAST. Okay, that's what we said last week, right? So we took our sequences, compared it by going to NCBI database, BLAST, and find exact match or any match. It searches, and it brings all previously reported TCDC genes. The full intact TCDC, the 18 base pair deletion, ours has 49 base pair. So it will show you this sequence match, and then dot. That is a deletion mutation, which was longer in our case, 39, and then matching continues. That's how you do it. So.
I have just one small question.
Okay, very good. Go ahead, yeah, please.